So we're really interested in cholesterol and cholesterol metabolism. So I just start with a disclosure that WG and YW are listed as inventors on the patent method for the quantitative detection of steroids, and we're also shareholders in a company called Calisthenics. Okay, so sort of an overview of the talk. So we'll be talking about cholesterol metabolism, and we'll look at the different pathways to bile acids. So the pathways that we're all familiar with, and also some pathways which are more unusual. I'll tell you about the methods that we use, which are based on LCMS. Then we'll just look at two inborn errors of cholesterol metabolism in a little bit more detail. And then I'll sort of, towards the end, last 5 minutes or so, I'll sort of pose the question, is Huntington's disease an inborn error of metabolism? And then we'll just, I'll just sum up. Okay, so it's cholesterol is the molecule that we are really interested in. Yeah, okay. So my interest in cholesterol and cholesterol metabolism really dates back to the late 1980s, when I was really lucky to actually go to Karolinska Institute in Stockholm and work with this chap. This is Jan Scherval, and he's probably the greatest living expert on cholesterol metabolism and bile acids. So he's a clinician, but really fascinated by analytical chemistry. And I guess he's probably the cleverest guy I've ever met. So I was really lucky to actually be able to work with Jan for actually quite a long time. So we were in Karolinska, and we were in the campus in Solna, which is quite near to the city centre. And then there's a second campus in Karolinska, which is in Huddinge, which is a southern suburb of Stockholm. And in Huddinge, there was Ingmar Björkem, and Ingmar Björkem is also a great expert on bile acids. But while Jan was interested in the whole pathway of cholesterol all the way to bile acids and the different bile acids, Ingmar was more focused on the very early steps of cholesterol metabolism. So Ingmar was there in Huddinge, and Jan was there in Karolinska, and I was exposed to both of them. So this is really lucky. So both Jan and Ingmar were interested in how bile acid pathways are affected in different cholesterol-related rare diseases. So they were both interested in Niemann-Pick disease, and also both interested in cerebrotendinous xanthomatosis, or CTX, which we'll come to later. So not only were they interested in rare disease of cholesterol metabolism, but Ingmar was really interested also in how cholesterol metabolism might be involved in neurodegeneration. So he was sort of interested in the more popular areas of the scientific subject. So I was really lucky to work with these guys. So then I came back to the UK in 2003, actually to the School of Pharmacy just across the road from Great Ormond Street, and worked there for I think 5 years, and then moved down to Swansea. And in Swansea, we've concentrated really all our efforts looking at different pathways of cholesterol metabolism. So we've been quite lucky to get funding from UKRI to actually develop methods. And then once we have the methods developed, we've been again lucky to get funding from large US charities, Michael J. Fox Foundation and the CHDI Foundation, who funded us to look at cholesterol metabolism in Parkinson's disease and also in Huntington's disease. And then, as Wendy mentioned, now we've got money from MRC to be a rare disease research node. So our node is lipidomics and metabolomics for rare disease, and this is a node of Swansea, UCL, Cardiff, and Manchester. Okay, that's a sort of a general introduction of how we got here. Now let's look at some science. So this is what we're interested in: cholesterol metabolism. Now, the first step of all cholesterol metabolism is to form an oxysterol. So oxysterols are oxidized forms of cholesterol. So this is the first step in all of cholesterol metabolism. And if you look in blood plasma, you'll see lots of different oxysterols, and these are the main ones that you'll actually see. So this one is 24-hydroxycholesterol. So the name comes from the fact that there's a hydroxy group at position 24. So this is an interesting oxysterol that we'll talk about a little bit later, in that it's only made in the brain. It's made in neurons, and it's made by the enzyme CYP46A1, which is only expressed in neurons. So all the 24-hydroxycholesterol that we see in our blood actually comes from the brain. Okay, next to that, we've got 25-hydroxycholesterol. And as the name suggests, we've got a hydroxyl group at position 25. And this is, again, an interesting metabolite, as it seems to be only formed by activated immune cells. So it's usually a minor oxysterol, but if you have a bacterial or viral infection, the levels can shoot up. Next, we've got 26-hydroxycholesterol, also called 27-hydroxycholesterol, and this seems to be pretty ubiquitous, and it seems to be made by most cells. So hydroxy group at position 26. Next, we've got 7-alpha-hydroxycholesterol, hydroxy at 7-alpha position. So this is made in the liver, because the enzyme that makes it, CYP7A1, this is liver specific. And then the next three I've got here: 7-beta-hydroxycholesterol, 7-ketocholesterol, sometimes I call this 7-oxocholesterol, and this cholestyne triol. So these are interesting oxysterols, as they're formed via non-enzymatic reactions. So normally, again, these are quite minor, but in certain situations, certain disease states, these shoot up. Now, all of these different oxysterols can be converted to bile acids, and this is something that we've spent the last 15 years looking at the different pathways that each of these different oxysterols may have to end up as bile acids. If we look at 26-hydroxycholesterol and 7-alpha-hydroxycholesterol, the pathways of these two molecules to bile acids are well established, already very well known. So here's the conventional pathways, or the normal pathways to bile acids that we normally see in the healthy state. So here's cholesterol, and then there are actually two main pathways. There's what we call the acidic pathway, which starts with this CYP27A1 making 27-hydroxycholesterol, and then making an acid, hence the name of the acidic pathway. And the acidic pathway, we have side chain shortening, reversing of the stereochemistry at position 3, and saturation of the double bond, and we end up with keno-deoxycholic acid. So most of the keno is formed via this acidic pathway. Now, the acidic pathway, the first early steps can be non-hepatic. So many of these early metabolites are present in blood, formed by non-hepatic cells. On the other hand, the neutral pathway, this is a hepatic pathway, and this pathway starts with CYP7A1 introducing a 7-alpha-hydroxy group into cholesterol to give a 7-alpha-hydroxycholesterol. So CYP7A1 is only expressed in the liver. So this pathway is liver specific, and then we end up with cholic acid. The pathways can intercross, so you can get one. you can get the same metabolites in both pathways. So this is actually very well established biochemistry, textbook biochemistry. So as I said, our interest really has been in the pathways from these other metabolites, which in most cases are probably not important, but in certain cases they will be important. So, for example, if the conventional pathways aren't working, we may have metabolism shifted into an unusual pathway. Or alternatively, perhaps if we've got very high levels of 25-hydroxycholesterol as a result of inflammation, we may have metabolism shifted into a metabolic pathway dominated by 24-hydroxycholesterol. On the other hand, certain diseases, for example, the Niemann-Pick and the Wallman diseases, we seem to get a lot of non-enzymatic formation of these oxysterols, and then these fall into, again, different pathways to generate different bile acids. So this has sort of been our interest for the last number of years. We're also interested in the precursors of cholesterol. As actually the precursors of cholesterol, they can also be converted into bile acids. So, for example, in the disorder Smith-Lemli-Opitz syndrome, where this enzyme 70-hydrocholesterol reductase is deficient, you get very high levels of 70-hydrocholesterol. And 70-hydrocholesterol has got its own pathway to give bile acids, which actually goes through 7-oxocholesterol, also called 7-ketocholesterol. So what you're probably thinking now is what's the point in these people studying these unusual metabolic pathways? Well, I guess the most obvious reason is that high levels of unusual metabolites in these pathways can be actually diagnostic for different metabolic disorders. So strange metabolites in our plasma can help in diagnosis. For many of the inborn errors of cholesterol metabolism, there are already good biochemical markers. However, some of these markers may actually be less good when we have late onset inborn errors of metabolism, where perhaps an enzyme which has only got partial activity. So in this case, some of the conventional biomarkers may actually be less good. So these unusual markers may come into their element. Also, by observing strange pathways, then we observe the buildup of unusual molecules. These may tell us about the pathology of different diseases. So a particular molecule that builds up may be toxic, or on the other hand, a particular molecule that is no longer formed, that might be protective, and we lose a protective effect. And finally, a good reason to monitor or to analyze these unusual metabolites is that we can look at them to monitor the response to therapy. And at the end, I'll show you how this can be used for looking at Wallman disease. Okay, so we're interested in all these cholesterol metabolites. So we use LCMS. And a disadvantage really of LCMS for the cholesterol precursors and the early cholesterol metabolites is that these molecules give pretty poor signal in conventional LCMS. So really inspired by earlier workers, we sort of developed a derivatization method to improve the ionization of these early metabolites to make them more visible in our LCMS methods. So this is our derivatization strategy. It's a two-step derivatization strategy, which we call enzyme-assisted derivatization for sterol analysis. And it's dead simple. So most of the early metabolites of cholesterol and the cholesterol precursors have got this 3-beta hydroxy group. So in a first step, we convert the 3-beta hydroxy group to a 3-oxo group or a 3-keto group. So we do this using cholesterol oxidase enzyme. So you can just buy bacterial cholesterol oxidase enzyme from Sigma, add it to your extract, and you'll convert the 3-beta hydroxy group to a 3-oxo group. Now, 3-oxo groups or keto groups, these are good targets for further chemistry. And what we can do is we can use this reagent. It's called the Girard reagent, and the Girard reagent's got this hydrazine group. And hydrazine groups will react with ketones through very classical chemistry to form a hydrazone. So if you add this one to the ketone, you click the Girard hydrazine onto the steroid. When you do that, it makes the steroid light up in the mass spectrometer. The reason for this is that we're adding a permanent positive charge. So now the molecule lights up in the mass spectrometer, and we had a great improvement in sensitivity. So the chemistry here is really mature. So this is classical Richmond chemistry used in measurement of cholesterol, and this classical hydrazine reaction. Again, it goes back to Girard in 1936. So the chemistry we're using is really robust. So a little cartoon of what happens. So conventional oxysterol, conventional early cholesterol metabolite, it's got this 3-beta hydroxy group, pretty poor signal. We convert that to the 3-oxo group. Once we got the 3-oxo group, we add the Girard reagent. Girard reagent clicks onto the oxo group. Up goes the signal by a factor of 100 or 1,000, depending on your mass spectrometer. So this is really nice and easy, very simple derivatization chemistry. An added advantage of the derivatization is that when we go to fragment our derivatized molecules, they fragment in a really nice way. So we get this really informative fragmentation in the ring system and also in the side chain. So that means that if we get an unexpected peak in our LCMS chromatogram, we can fragment it. And from the fragmentation pattern, we can identify what cholesterol metabolite we're actually looking at. So just a couple of fragmentation patterns. This is 24-hydroxycholesterol, and this is 27-hydroxycholesterol. So the patterns are similar because, of course, the molecules are similar, but they're different in that 24-hydroxycholesterol has these peaks at 325 and 353, while the 27-hydroxycholesterol, these peaks are really minor. And also we see a reversal in the intensities of these high mass fragments between the two molecules. So the fragmentation patterns are actually really useful. Okay, the derivatization doesn't work on all cholesterol metabolites. So, the primary bile acids, they don't have a 3-beta hydroxy group. Neither do they have a 3-oxo group that we could react with. So these are transparent to our derivatization. However, the primary bile acids, and actually the late steps of the bile acid biosynthesis pathway, they've got a carboxylic acid group. And of course, carboxylic acid groups really give good ions if we just flip our mass spectrometer from positive to negative ion analysis. So if we're interested in the bile acids themselves, we can just flip our mass spectrometer from the positive ion mode to the negative ion mode. So in fact, if we look at our normal bile acid biosynthesis pathways, using our derivatization and then flip it into negative ion mode at the end, we can actually observe just about all the metabolites in the bile acid biosynthesis pathway. Okay, while I've got this pathway up here, let me sort of concentrate again a little bit more on this acidic pathway. Now, the first enzyme in this acidic pathway is called CYP27A1. And CYP27A1 converts cholesterol into this 27-hydroxycholesterol, and it then converts the 27-hydroxycholesterol into a carboxylic acid, a C27 carboxylic acid. Now, there's a disorder called CTX, or cerebrotendinous xanthomatosis, where CYP27A1 doesn't work. So this disorder, CTX, cerebrotendinous xanthomatosis, so that's a deficiency in the CYP27A1 enzyme. So this CTX, it can present with cholestatic liver disease in infants. It can present with juvenile cataracts, and then more in the adult, you get a decline in mental function, and you also get these tendinous xanthomatosis. However, once you diagnose CTX, there's a excellent treatment which is actually chenodeoxycholic acid itself. And Peter Clayton, who's here today, he's seen many cases of CTX, and he's treated them with cheno and with excellent outcomes. So these CTX people with CTX, because the CYP27A1 is not working, the acidic pathway is essentially turned off. So 27-hydroxycholesterol isn't made, and neither are any of these acids. So the acidic pathway is turned off. Metabolism is sort of shifted into the neutral pathway. However, CYP27A1 also appears in the middle of the neutral pathway. So what this means is the neutral pathway is blocked. So we've got a shift into metabolism of the neutral pathway, but the pathway is blocked in the middle. So the result is a huge increase in the early metabolites of the neutral pathway, particularly this one, which is 7-alpha 12-alpha dihydroxycholesterol. 7-alpha 12-alpha dihydroxycholesterol. So what if you take a drop of blood from a CTX patient, what you'll see is the 7-alpha 12-alpha dihydroxycholesterol shoots up, while 27-hydroxycholesterol and the acids come down. And we can observe these all using our simple derivatization method. So just show you some data. So normally this 7-alpha 12-alpha dihydroxycholesterol, this is really minor metabolite. So what I'm showing you here is we've got controls in blue, CTX on treatment in red, CTX without treatment in green, and CTX, well, where we didn't know whether they were treated or not treated. And here we've actually got a log scale. So the CTX patients, those which are untreated, they've got about 1,000-fold more 7-alpha 12-alpha dihydroxycholesterol than our controls. On the other hand, when we look at 27-hydroxycholesterol and the downstream acids, so 27-hydroxycholesterol, this is normally about 40 nanograms per mil, but in CTX, it can be 1 nanogram per mil or less. So 7-alpha 12-alpha dihydroxycholesterol goes up, 27-hydroxycholesterol and the acids go down. So really, to get the best diagnostic, if we actually look at some ratios, so if we work out the ratio of 7-alpha 12-alpha dihydroxycholesterol to 27-hydroxycholesterol, 7-alpha 12-alpha dihydroxycholesterol has gone up, 27-hydroxycholesterol has gone down, then the ratio actually becomes massive in CTX. Well, very, very minor in the control. So we got really, by looking at the ratios of the 7-alpha 12-alpha dihydroxycholesterol to either 27-hydroxycholesterol or the acids, you really have a incredibly good diagnostic. So you got a 10,000-fold increase in the ratio in CTX. So this is really neat. Of course, there are other diagnostics for CTX, and probably of the others, probably the best is the cholestain cholestein tetralglucuronides, bile alcohol glucuronides. So these go up in CTX. So these also provide a good diagnostic for CTX, and we can measure them just as easily in our method because they give nice ions in the negative ion mode. And also cholestanol, this is formed via a shunt pathway. This also goes up in CTX. But perhaps a drawback of these two is that cholestanol, neither cholestanol or cholestein tetralglucuronide, their elevation is not specific to CTX. So the glucuronides can also be high in other inborn areas of metabolism, but particularly the peroxisomal disorders, while cholestanol can be elevated in phytosterolemia. So these are perhaps less specific. So while these will provide you with a very good indication that you've got CTX or not, our ratios will actually could then go on and give you a definitive diagnosis. Let me go back a slide. So in this, if we go back to the acidic pathway again, in the acidic pathway, we get this 27-hydroxycholesterol. And 27-hydroxycholesterol, when we introduce this hydroxy group at position 27, we introduce stereochemistry at this asymmetric center. And CYP27A1 always introduces R stereochemistry. So when we were analyzing our CTX patients, surprisingly, we found some metabolites which actually had S stereochemistry. So this was really interesting. So what we actually found out by actually doing some enzymatic studies is that there's a second sterile 27 hydroxylase other than CYP27A1, which actually introduces 25R stereochemistry, 25S stereochemistry rather than 25R stereochemistry. This is actually CYP3A4. So what actually we find is that in the neutral pathway, some of that 7-alpha hydroxycholesterol, which is formed in the first step, can be converted by CYP3A4 to it can be 27 hydroxylated and then converted to an acid. And both of these have S stereochemistry rather than normal R stereochemistry. And these will then be converted to quinodeoxycholic acid. So in people with CTX, if CYP27A1 was absent, you'd predict there'd be no quino. However, very often you see a small amount of quino, and almost certainly the small amount of quino is actually generated via this unusual pathway, which is involved with CYP3A4 introducing and this 25S stereochemistry to 27 hydroxycholesterol. So this is one of the pathways that we sort of have been able to find by analyzing all these cholesterol metabolites in detail. Okay, so there we were looking really at the well-established pathways and how they change in CTX. Now let's change track and look at some bile acid biosynthesis pathways from some other cholesterol metabolites. And let's start first. Let's look at 25-hydroxycholesterol. So this 25-hydroxycholesterol, as I said, this is a very interesting oxysterol in that it's normally in plasma. It's really a minor oxysterol. However, it's made by activated immune cells. So as a result of inflammation, as a result of infection by bacteria or viruses, CH25H, the enzyme that makes it, is upregulated, and you get great increases in 25-hydroxycholesterol. And it's thought that probably 25-hydroxycholesterol is actually an immuno-oxysterol and probably is protecting us from both bacterial and viral infection. So it must have been about almost 10 years ago, maybe 8 years ago, we were contacted actually by Simon Jones's team in Manchester, and they'd come across some really interesting patients who were deficient in cholesterol 25 hydroxylase. So these patients were very unusual in that they had a deletion in the LIPA gene. This is the gene which codes for lysosomal acid lipase. So they were deficient in LIPA, and also the next gene along, which is cholesterol 25 hydroxylase. So these were really unusual patients who had this double deletion. And what Simon's team found is that these patients seem to present with BCG abscesses. So it's, my thought is probably the absence of 25-hydroxycholesterol in these patients is probably responsible for these abscesses. Anyway, so Simon's team sent us some plasma from these patients, really to confirm the absence of 25-hydroxycholesterol. So at the bottom, I've got a normal chromatogram of normal plasma. And 25-hydroxycholesterol, as I said, in normal plasma, it's pretty minor, but you can see there's the peak. It's a clear peak. But when we got the plasma from these patients, absolutely nothing there. No 25-hydroxycholesterol was being produced by these patients. So that really confirmed that the double deletion made the patients made an absence of 25 hydroxycholesterol. So these patients are actually really interesting patients in that they've got the double deletion and the LIPA deletion, that gene before cholesterol 25 hydroxylase. Deletion in that gene results in Wallman disease. So what we've got in Wallman's disease, so we've got deficiency in the enzyme lysosomal acid lipase in the lysosome. So lysosomal acid lipase hydrolyzes cholesterol esters into cholesterol. So these patients, they've got a error in the hydrolysis of cholesterol esters, and also they've got the deficiency in cholesterol 25 hydroxylase. So they've got really complicated phenotype. And what happens with this Wallman disease, when we have the deficiency in the lysosomal acid lipase, that means that the cholesterol is not released as normal in the lysosome. And cholesterol actually provides a negative feedback mechanism on its own synthesis. So what's probably happening in Wallman's disease per se is that you've got a loss of negative feedback on cholesterol biosynthesis. So probably in the Wallman's disease, you've probably got an overproduction of cholesterol in the cells via endogenous synthesis. So, okay, so Wallman's disease, it can be treated by enzyme replacement therapy. However, if the symptoms deteriorate, then the treatment is hematopoietic stem cell transplant. So there were 3 patients found with at least 3 patients found with this double deletion of LIPA and CH25H. And these 3 all had the hematopoietic stem cell transplantation. So Simon's team sent us down samples of plasma from these patients to actually monitor really the response to therapy. So if we look at a patient before treatment, we see no 25-hydroxycholesterol, but we do see these strange cholesterol metabolites here. These are formed as a consequence of the Wallman disease per se. So each of the patients then had the transplant, and after the transplants, they're transplanted with cells from healthy donors. So they're transplanted with the cholesterol 25 hydroxylase gene, and also the lipid gene. So naturally up goes the 25-hydroxycholesterol level. So 4 months after transplant, 25-hydroxycholesterol goes up. Then as time goes by, the levels of 25-hydroxycholesterol drop down to normal, and also these other metabolites drop down to normal. So all three patients that we've looked at now, they're all surviving, and the levels of the metabolites all look quite normal. So when we look at these patients, not only did we look at 25-hydroxycholesterol, but we also looked at all the other metabolites we could actually see. So here we're plotting actually how different metabolites vary with time. So in the white, it's pre-treatment. In the gray, it's after treatment. And at the far side here, we've got data for the mother and father. So 25-hydroxycholesterol is in blue. So before treatment, no 25-hydroxycholesterol. After treatment, 25-hydroxycholesterol shoots up. So that's true in the three patients. It then drops down to normal levels, and that's true in each of the patients. So if we look at one patient in a little bit more detail, so not only does 25-hydroxycholesterol shoot up, but other metabolites also shoot up at the same time point. And these other metabolites in green, brown, and orange, these are all derived from 25-hydroxycholesterol. So the green one is 7-alpha hydroxylated. It's got 25-hydroxy group, but it's also got an acid group. The brown one is very similar, but it's got a 3-ketone group. And the orange one, again, is very similar, but instead of having a 7-alpha hydroxy group, it's got a 7-beta hydroxy group. So basically, what we're actually observing here is that after transplant, you get these super high levels of 25-hydroxycholesterol. And when you get the super high levels of 25-hydroxycholesterol, then we see a pathway of metabolism shifted, but basically a new pathway of metabolism shifted where we get these acids formed, which no doubt go on to actually ultimately give us bile acids. So looking at this, looking at these patients, we start off with no 25-hydroxycholesterol, transplant high levels of 25-hydroxycholesterol, and then we see a new pathway of bile acid biosynthesis. Okay, I'll just take 5 minutes more of your time to tell you a little bit about some work we've done on Huntington's disease, and I just throw out this sort of almost jokey question: is Huntington's disease an inborn error of metabolism? So first of all, a few words about Huntington's disease. So it's really a horrible disease. So it's an autosomal dominant neurodegenerative disease. It results from a CAG expansion in the Huntington's disease, in the Huntington's gene. So normal people have a CAG repeat of about 27 units, but people who will get fully penetrant Huntington's disease will have a CAG repeat of 40. And it's a horrible disease because it's autosomal dominant, and it presents, in middle age. So you'll probably know that you're going to get it because one of your parents did, and it's going to come to you in middle age. So it's a terrible disease, and there's no treatment. So why do I say, is it an inborn error of metabolism? Well, the mutant Huntington with these extra repeats, there's pretty good data that it inhibits cholesterol synthesis and also modulates or changes cholesterol metabolism. So clearly, in Huntington's disease, cholesterol synthesis or metabolism is being affected, and it's being affected by this mutant Huntington protein. And there's sort of two groups of thought of how mutant Huntington's can actually be causing the pathology of Huntington's. So one idea is that it's really the pathology of Huntington's is a consequence of really cholesterol deficiency in the brain, particularly in the striatum, where Huntington's disease kills off the neurons most clearly. So that's one idea, and this idea comes from a group in Milan, Valenza and Cataneo. And then there's another idea that really that the mutant Huntington is not causing a defect in cholesterol synthesis per se, it's actually causing a defect in cholesterol metabolism. So there's a French group who think that perhaps a good treatment of Huntington's disease might be trying to increase cholesterol metabolism, while the Italian group think a good treatment of Huntington's disease might be to increase cholesterol biosynthesis. So you've got these two ideas of treatments of Huntington's disease, one focusing on synthesis and one focusing on metabolism. So we got interested really in Huntington's disease, really because we can measure 24-hydroxycholesterol. And if you remember, I told you at the very beginning, the 24-hydroxycholesterol is only made in brain. So, and it's only made in neurons, because neurons express the enzyme CYP46A1, and only neurons express it. So all the 24-hydroxycholesterol we have in our plasma all comes from brain. So we thought perhaps by measuring 24-hydroxycholesterol levels in plasma from HD patients, maybe we might have a biomarker for the progression of the disorder, and maybe it could help solve the problem of who's got the best treatment. Is the best treatment increasing cholesterol synthesis, as suggested by the Milan group, or is the best treatment going to be increasing cholesterol metabolism, as suggested by the Paris group? So we were funded by this CHDI, rather wealthy US charity, and they actually collected all the HD samples, or they funded the collection of all the HD samples. And I think they gave us something like 400 samples. So we had healthy controls in blue here, pre-manifest HD. So that's before the onset of the the symptoms of HD, and then samples from people with bona fide AHD itself. So we analysed both plasma and CSF, and actually the plasma data is actually the most interesting. So when we looked at the data, the levels of 24-hydroxycholesterol in the manifest HD, the plasma of manifest HD, is statistically lower than in either controls or in the pre-manifest. When we then sort of did some, or when we actually gave the data to a statistician to actually model, he then was able to take into account the effects of sex and the effects of age. And when you take into account age and sex, and then look at 24-hydroxycholesterol levels, here we're actually plotting log, it's actually natural log concentrations of 24-hydroxycholesterol against age. And manifest HD is red, pre-manifest is green, and controls are blue. You can see whatever the age is, the levels of 24-hydroxycholesterol in plasma from both males and females is lower in the manifest state than the previous than in the pre-manifest state or in the controls. So certainly 24-hydroxycholesterol is affected in Huntington's disease. So maybe it is an inborn error of metabolism. Maybe that mutant Huntington is affecting the metabolism of cholesterol to 24-hydroxycholesterol. However, actually, that's not the only explanation. So 24 is definitely diminished in plasma from manifest HD patients. So 24 has definitely gone down in manifest HD patients. And 24, it's got to be made from cholesterol, and it's got to be made from CYP46A1, and this has got to happen in brain. So maybe it's gone down because of reduced availability or reduced function of CYP46A1. So this would fit the hypothesis from the French group. So maybe 24 has gone down because there's defect in metabolism. However, that's not necessarily so. Maybe 24-hydroxycholesterol has gone down because there's a decreased availability of the substrate, i.e. cholesterol. So maybe the 24-hydroxycholesterol has gone down because we've got less cholesterol, which actually would fit the Italian hypothesis. So our study really couldn't answer the question, but it can tell us that 24-hydroxycholesterol can be a biomarker for HD, which of course can be useful in the development of therapies. And as I said, there are no good therapies at the moment. So finally, is a summary of what I said here. So cholesterol is metabolized by multiple pathways. Pathways might be minor in the healthy state, but can become major in diseases. And elevated or diminished pathway metabolites, that can perhaps tell us more about the pathology. And then finally, cholesterol metabolites can be biomarkers for perhaps the common diseases, perhaps Parkinson's disease, and also the rare diseases like those of inborn errors of cholesterol metabolism. So finally, the acknowledgement slide. So this is Yutin, who run the lab. Most of the work's been done by Mosin and Elan. Manuela and Lauren have also been great help. Statistics for the HD was Doug Langham. Funding for HD was CHDI. We get help from the Welsh government. This used to be when we were part of the EU, so this is all dried up. We've had help from UKRI, and now we're part of this rare disease node, and that's the end. Thank you very much for listening.